Every second of your life, you are under attack. Billions of bacteria, viruses, and fungi are trying to make you their home. So our bodies have developed a super complex little army with guards, soldiers, intelligence, weapons factories, and communicators to protect you from, well, dying. For this video, let's assume the immune system has 12 different jobs. For example, kill enemies, communicate, etc. And it has 21 different cells and 2 protein forces. These cells have up to 4 different jobs. Let's assign them. Here are the interactions. Now, let's make this understandable. First of all, let's add colors to the jobs. Now, let's illustrate the cells. The central color represents the main job of the cell, while the surrounding ones represent secondary duties. Now, the immune system looks like this. Now, the interactions. Isn't this complexity just awesome? For this video, we will only talk about these cells and ignore the rest. So, what happens in the case of an infection? It's a beautiful day when suddenly a wild rusty nail appears and you cut yourself. The first barrier of the immune system is breached, your skin. Nearby bacteria seize on the opportunity and enter your wound. They start using up the body's resources and double their numbers about every 20 minutes. At first, they fly under the radar, but when a certain bacteria population is reached, they change their behavior and start to damage the body by changing the environment around them. The immune system has to stop them as fast as possible. First of all, your guard cells, known as macrophages, intervene. They are huge cells that guard every border region of the body. Most of the time, they alone can suffocate an attack because they can devour up to 100 intruders each. They swallow the intruder whole and trap it inside a membrane. Then the enemy gets broken down by enzymes and is killed. On top of that, they cause inflammation by ordering the blood vessels to release water into the battlefield, so fighting becomes easier. You notice this as a very mild swelling. When the macrophages fight for too long, they call in heavy backup by releasing messenger proteins that communicate location and urgency. Neutrophils leave their patrol routes in the blood and move to the battlefield. The neutrophils fight so furiously that they kill healthy cells in the process. On top of that, they generate barriers that trap and kill the bacteria. They are indeed so deadly that they evolved to commit suicide after 5 days to prevent them from causing too much damage. If this is not enough to stop the invasion, the brain of the immune system kicks in. The dendritic cell gets active. It reacts to the signals of the soldiers and starts collecting samples from the enemies. They rip them into pieces and present the parts on their outer layer. Now, the dendritic cell makes a crucial decision. Should they call for antivirus forces that eradicate infected body cells or an army of bacteria killers? In this case, antibacteria forces are necessary. It then travels to the closest lymph node in about a day. Here, billions of helper and killer T cells are waiting to be activated. When T cells are born, they go through a difficult and complicated training process, and only a quarter survives. The surviving cells are equipped with a specific setup. And the dendritic cell is on its way looking for a helper T cell with a setup that's just right. It's looking for a helper T cell that can bind to the parts of the intruders which the dendritic cell has presented on its membrane. When it finally finds one, a chain reaction takes place. The helper T cell is activated. It quickly duplicates thousands of times. Some become memory T cells that stay in the lymph node and will make you practically immune against this enemy. Some travel to the field of battle to help out. And the third group goes on to travel to the center of the lymph node to activate a very powerful weapons factory. Like the T cells, they're born with a specific setup. And when a B cell and a T cell with the same setup meet, hell breaks loose. The B cell duplicates rapidly and starts producing millions of little weapons. They work so hard that they would literally die from exhaustion very fast. Here, helper T cells play another important role. They stimulate the hard-working factories and tell them, "Don't die yet, we still need you. Keep going." This also ensures that the factories die if the infection is over, so the body doesn't waste energy or hurt itself. But what is produced by the B cells? You've heard of them, of course, antibodies. Little proteins that are engineered to bind to the surface of the specific intruder. There are even different kinds of antibodies that have slightly different jobs. The helper T cells tell the plasma cells which type is needed the most in this particular invasion. Millions of them flood the blood and saturate the body. Meanwhile, at the site of infection, the situation is getting dire. the intruders have multiplied in number and start hurting the body. Guard and attack cells fight hard, but also die in the process. Helper T cells support them by ordering them to be more aggressive and to stay alive longer. But without help, they can't overwhelm the bacteria. But now the second line of defense arrives. Billions of antibodies flood the battlefield and disable lots of the intruders, rendering them helpless or killing them in the process. They also stun the bacteria and make them an easy target. Their back is built to connect to killer cells, so they can connect and kill the enemy more easily. Macrophages are especially good at gnoming up the bacteria which antibodies have attached to. Now, the balance shifts. In a team effort, the infection is wiped out. At this point, millions of body cells have already died. No big deal, the losses are quickly replenished. Most immune cells are now useless, and without the constant signals, they commit suicide, so as not to waste any resources. But some stay behind, the memory cells. If this enemy is encountered ever again in the future, they will be ready for it and probably kill it before you even notice. This was a very simplified explanation of parts of the immune system at work. Can you imagine how complex this system is? Even at this level, when we ignore so many players and all the chemistry. Life is awfully complicated, but if we take the time to understand it, we always encounter endless wonders and great beauty.
So this week we're covering immunology. Let's get into it. So the history of immunology is really tied to the history of infectious diseases. We've always known that people can get sick with something. A lot of people get hurt and die from those things, but some people recover. And that's kind of the key thing to immunology, is that we can recover. We have some kind of memory inside of our bodies that remembers infectious diseases. And the first person to note that was I don't know how to say his name right. Thucydides. And he was way back in BC, and he noticed he was in Athens, and that when Athenians were recovering from plague, so they called it plague back then, but it probably wasn't actually plague, which is a bacteria. It was probably smallpox, most likely, or maybe measles, which those are viruses, and we're going to actually talk next week about viruses, and it's going to be really cool, and we'll talk a little bit more about infectious disease history next week. But the idea that someone could recover from a disease, that the same man is never attacked twice, is the quote that's given to him, and that's immune memory, and we're going to talk about that today. This is also tied a lot to vaccines, which we don't have time to cover this week, and so we're going to cover it next week when we also talk about viruses. But the idea of taking dried pieces of pus, which sounds super gross, from smallpox. So smallpox is a virus that we actually eradicated from the globe. And we can take little pieces, or they used to take little pieces of powdered smallpox stuff and shoot it into your nose, or maybe scratch it into your arm. And that's called variolation. And that idea of introducing an infectious agent into a person to prevent future infections. So that's why we understand now, like, the immune system remembers stuff. And it really was smallpox that was the one that really drove our studies of immunology. And so for a long time, we knew, okay, if we expose people, which is kind of like vaccination, it's a little bit different, but if we expose people to an infectious agent, they'll have immune memory. And so hundreds and hundreds of years pass, and we just started to notice that we could keep on doing this. And so variolation was really popular. Smallpox was just killing tens, hundreds of millions of people. And so we knew, okay, there's some folklore. Nobody was really studying this super well. They said, okay, we know that people can get sick and then not get sick again with the exact same thing. And so there was some folklore to that. And there was a guy named Benjamin Jesty, and he noticed that on his farm, he would have milkmaids that would milk the cows. And he noticed that they would get this disease called cowpox, which it's like this little pock-marky looking thing. It kind of looks like this. And they would get cowpox, and then they wouldn't get smallpox, which smallpox would just decimate an entire village, except for the milkmaids. And so he noticed that, hey, they were kind of immune. I mean, he didn't know it at the time. And so what he did was he purposely infected his kids, and I think his wife too, with cowpox, and then they didn't get smallpox when it came through the town and wiped out everybody else. But Jenner is actually the one who is considered the father of modern immunology. And he actually kind of was more meticulous and scientific about actually studying the idea of giving people cowpox to make them immune to smallpox. And basically what happened was those viruses are really similar in some of their structures and some of their surface proteins, which we'll talk about next week. And it was kind of having this cross immunity between those two. And cowpox doesn't hurt people as much. Variolation was actually using the original smallpox, which would make people really sick. That's not ideal. You don't want to give people smallpox. And so they found this alternative. Now, that vaccine was awesome. The idea of actually giving people cowpox to prevent smallpox, it saved a lot of people's lives. And so a lot of people started working even more and trying to figure out, exactly what's happening, because they didn't know physically what was happening. They just knew, like, hey, we're seeing some kind of memory, like your body is remembering these infectious agents. And so cut to about 100 years later-ish, Louis Pasteur was around, and we've heard of him before, pasteurization. He was just a hardcore scientist. He was doing all kinds of stuff, and he was working on a bunch of different vaccines because that was super in at the time. So working on something for chickens. We don't usually think about vaccines for animals like chickens and fish and dogs and things like that, but there are a lot of vaccines for those because those are huge industries. So he was learning a little bit kind of the tricks of the trade with immunology, the difference between viruses and bacteria, which that wasn't really well understood at the time. What really put Pasteur on the map and what really put vaccines on the map at the time, I mean, it was helping with smallpox, but he had developed a rabies vaccine, which rabies is one of the deadliest viruses we know. And an 8-year-old boy got bit by a rabid dog. They knew he was going to have rabies. And so they were able to give him some vaccine, and he survived. And that was huge. It made international headlines. It was like a huge deal. And so you can see the cart is kind of coming before the horse. Like, we understand how to get immune memory into people, but we don't actually understand what's going on. And so that actually took a few years, maybe a few decades really, to get better technology, basically. And so we're going to encounter Paul Ehrlich now. So Ehrlich had a friendly rivalry with a guy named Mechnikov. And these two dudes are really more the fathers of immunology. Maybe they're the godfathers of immunology. And so Ehrlich he had this idea. We knew that you could take someone's serum out. And so you separate all the cells of the blood out, and you just have the serum left over. And they knew that if you took some of that serum and you injected it into somebody, you could actually stop an infection. And so what he demonstrated, we called that humoral immunity. They knew there was something in the blood, but they knew there were no cells in the blood, and so they didn't know what that was. So they called it humoral after, the four-humor hypothesis from back in the day. They just knew it was something in the blood, and they didn't know what it was. Now, Mechnikov thought that it was cells. He thought the cells were the key to actually protecting someone from disease. And he would look at starfish macrophages. So he'd take larvae from starfishes. Then he would inject like weird stuff, like just, random junk inside these cells. And then these crazy looking cells would come out, and they would eat those bad things. And we're going to talk about what those are today. Those are called macrophages. And so they kind of had this friendly rivalry, but it turns out they were both right, and they both won the Nobel Prize in 1908 for their work. Now, there's a ton of history to immunology, and I have a really great article here. If you ever want to look it up, it's a free article, Frontiers in Immunology, Immunology's Coming of Age. It has the history from basically the earliest people all the way up until modern day. And I just have a little picture here. We're not going to go through all of these people, but all of them are really, really important and introduced really, really important ideas. But you can see there's Ehrlich, and they're spelling it Mechnikov, but it was Mechnikov too. It just kind of depends on what country you're in. And so that brings us to the immune system. We need to talk about it. Now, the immune system, we generally divide into two huge categories: the innate immune system and the adaptive immune system. These are sometimes called arms of the immune system. And so what we're going to do is we're just going to kind of go through. We're going to start with the innate immune system. We're going to start with our anatomical barriers, and then we're going to get into the cells of the immune system. And then we're going to spend a big chunk of time on the adaptive immune system and what that really means. So let's get into it first. So our anatomical barriers, we don't often think about them as part of the immune system, but if you don't have skin, then bad guys can just get in anywhere and kill you. That's one of the big problems with like a burn victim. You can get infections just into past that nice layer of skin cells because the skin cells are all gone, and then you just get a really bad infection. And actually, dehydration is one of the major causes of death for burn victims, but this is also a huge problem. And so we have lots of ways of preventing bad guys from getting in, of protecting the actual surface. So you have bacteria that live all over here, all over your skin, and so that's what I'm showing up here. These are just some of the bacteria. I mean, you have hundreds of types of bacteria that live on your skin. And so they are taking up space. They're eating things on your skin. That's where they live, and they want to protect where they live because, you want to protect where you live, so the bacteria protect where they live. There are also really, really tight junctions, so the intercellular junctions between all of your skin cells. So the skin is actually really complicated. It has a lot of different cell types, and they're really held together really, really tight. You also secrete all kinds of weird things onto the surface of your skin. So it's kind of acidic. You can think of sweat. Some of those dead skin cells on the surface of your skin can fall off. That would also keep things away. It's actually kind of dry on the surface of your skin. And you also secrete something called antimicrobial peptides. So they're called AMPs here. And so those antimicrobial peptides, I have a picture of a few of them here. Antimicrobial peptides are these really, really cool proteins that you secrete into various surfaces. So your skin, and your lungs, all kinds of places, basically any kind of surface. And they can just non-specifically kill certain bacteria. And so we have all kinds of different ones. So you can see there's a lot of variety in the shapes here. And they all have different names. But basically what they do is they kill specific types of bacteria that we know are dangerous. So if the skin faces the outside, we actually have a lot of other surfaces that face the outside as well. They're just inside of us, and we call those mucosa. And so the mucosa, so that's like the intestinal mucosa, the vaginal mucosa, the lung mucosa, the nasopharynx, basically anywhere that's kind of on the inside, but it's actually facing the outside world. And so those mucosal surfaces, we have lots of mucus on there. You can think your stomach is very acidic, right? There are lots of enzymes that we have there. So the same enzymes that we use to break down food can also kill bad guys that try and get into our stomach and our intestines. We also are able to expel bacteria and dirt and anything that gets into specific compartments. So like the vaginal compartment or the lungs, we have cilia. So you can see in the top right corner here, we have these cilia and they beat close together. And we've talked about cilia previously in this class when we talked about flagella and cilia and that combined movement. And what they can do is they can move that mucus back out. And so that's why, your nose will have the mucus start to come out, especially if you've been like in like a sooty area or like a smoky area, you'll have that mucus actually come out of your nose, or if you have a cold too. But that expulsion is really, really important and a great way to get things out of your body. Before I go into specific cell types, there were two big things that I did want to cover because we're going to spend a lot of time talking about immune cells. And so I wanted to get some things out of the way. And one of those is inflammation. It's super important that you understand what inflammation is. And so you could, of course, go very, very in depth in all of these things, but the signs of inflammation, and if you have an infection like on your finger or something, like what is actually happening in inflammation? Well, you're going to have your capillary beds start to widen. You have some leakiness start to occur, so that's permeability in your capillaries and all of your vessels, really. And you're able to attract immune cells, which we're going to talk about that. So leukocyte is just a generic name for a white blood cell, leuko meaning white. And so if you have a damaged area, so kind of imagine like if you stepped on a nail or something, or you cut your finger on like a rusty blade, then you want to get some nice white blood cells there to actually attack any bad guys that got in. And then also, like, let's say things are really, really not doing great. Well, then maybe you need to have a fever start to develop. You need to develop like a systemic response. And so these are just signs of inflammation that are expressed in the 4 or 5, depending on who you ask, cardinal signs of inflammation. So that's heat. So inflamed areas are warm because of all the increased blood flow. They can also turn red because there's a bunch more blood flow there, right? There's a bunch of red blood cells. It also starts to swell, so that vascular permeability, those little leaky spots between cells so that fluid can release into the tissues. That makes it easier for white blood cells to get out, but that also causes swelling. And then there's also a lot of pain involved. So everyone's had inflammation on their body in some way. And if you touch it, it's all of these things. It's warm, it's red, it's swollen, and it hurts. And those 4 things combined can make it... So imagine if it was your finger, then you don't want to use your finger anymore. And so the loss of function, which in this context, they're saying tenderness, but the loss of function is usually considered the 5th sign, the 5th cardinal sign of inflammation. But sometimes it's not included because it's more of a symptom of the other 4. But yeah. Now, jumping off of this last green box here on the far right, so the idea of a fever. So we generally have a fever occur if there's a prolonged infection. There's a variety of reasons that it might happen. But if you have a specific amount of inflammation, then what your immune cells do is they get kind of upset and they start secreting things that tells the rest of your body, hey, there's a really bad infection here. We need to kind of amp up the temperature of the body for a variety of reasons. Usually it's to suppress the growth of microbes to kind of give your body time to survive. Now, enzymes have a very specific range of temperatures, especially in bacteria, that they can function at. And so if you raise the temperature even by a couple degrees, that can have a really big impact on the growth and movement and metabolism of bacteria that your body is perfectly fine with, but bacteria really don't like. Now, that's where we have a connection between the immune system and your white blood cells and the things that they secrete. And so they can secrete these almost miniature hormones, not size-wise, but a smaller class of hormones called cytokines, cell kinds of these proteins that get secreted, and they're like mini hormones that go out into the body and tell it stuff. And so in this case, your white blood cells see a really bad infection, so they secrete these cytokines that cause inflammation and just tell the rest of the body that something bad is happening. And some of those can get to the hypothalamus and tell the hypothalamus to raise the body temperature. Now, that's a double-edged sword because we know that increased fever can also be really bad for your patient. And so we know it's kind of a tricky one. You have to be really careful with fevers. So that brings us to cells, and we're going to spend a lot of time talking about cells today because the cells of the immune system are super duper cool. The development of a white blood cell, or your red blood cells, but mostly we're talking about white blood cells here, from a stem cell all the way to whatever cell it's going to become, that's called hematopoiesis. So hematopoiesis occurs in the bone marrow, though some of the cells kind of finish out their life in different compartments, like the thymus. But for the most part, all of your white blood cells start out in the bone marrow. So you have what we call hematopoietic stem cells. And so those are stem cells that are able to develop into any of the white blood cells. And there's a couple further down the line. So you have your myeloid progenitors here, and you got your lymphoid progenitors, and those will just become specific cell types. So macrophages are one that we're going to talk about today, and those come from a myeloid progenitor, which came from a hematopoietic stem cell. And so you can see they just kind of slowly develop into these different things, and that's occurring in the bone marrow. And so what I want to do is talk about each of these specific cell types. And I want to start with neutrophils. So neutrophils are your most basic defensive cell. They're the most common leukocyte. So if you did a complete blood count on someone, they should have mostly neutrophils. That's your first line of defense. Pus is actually made of neutrophils. So when a neutrophil goes to a site of inflammation, it knows there's like a cut somewhere. The cells there are secreting these things that attract immune cells to them. And once the neutrophils are there, they're looking for bad guys. And what they can do is mostly they will degranulate. So if I back up just a little bit, so here we have these granulocytes here. So I labeled them granulocytes. So there's your neutrophil right there. And you can see there's little dots in there. So those are granules. And granules are these really cool little vesicles or little containers of weapons. And so they can be used to blow up bacteria or viruses or whatever you're encountering, or parasites. And they can poke holes in things. They can just do massive oxidation to bad guys. And so they're really, really powerful. And so we call that degranulation when a granulocyte secretes those things. Neutrophils are also capable of gobbling up bad guys. So that's a process called phagocytosis, which we usually kind of just think about with like macrophages being the ones that phagocytose, meaning big eater or cell eater. But neutrophils can also do that. And then there's something that was discovered relatively recently, only within the last couple decades, and that's called netosis. And NET stands for neutrophil extracellular trap. I know, it's a long word. But basically what neutrophils will do is they will explode and shoot out all of their nuclear material. So that's their chromatin, which is your DNA plus your histones. And if you didn't know this, DNA is really, really sticky. And so you can see here we have netosis occurring. So this neutrophil has sacrificed itself. It basically took its nucleus and exploded it everywhere. And so you can see it's all this weird, gross, these are histones it's supposed to be, and then DNA is the gray. And you can see all these little white pathogens are stuck in there. And it's a really, really cool weapon that neutrophils can use. Now, another granulocyte that you may have heard of is called an eosinophil. Its name comes from a specific stain called eosin, and it's very, very red. And so if you do a stain on someone's blood and you find a bunch of cells that look like this, if you have eosin in your stain, the granules stain really, really red. They're relatively uncommon unless you have a really bad allergy. then you will see lots of eosinophils. Or if you have a parasite, so like something really big like a helminth, like a worm, or like a tapeworm, or malaria, something like that. And you'll have lots of eosinophils, and their job is to actually blow up parasites. And so I have a really cool video here of a parasite being attacked by a bunch of eosinophils. They took a helminth and they stuck it in a petri dish and put a bunch of eosinophils in there, and then you can see they just swarm it and degranulate and kill that thing, or like crazy. Now, the other granulocytes are basophils and mast cells. And so for a class like this, we're just going to kind of lump them together because they do share a lot of similarities. Some people kind of associate them as the exact same thing. That's absolutely not true. But for the most part, their job is sort of antiparasitic. They are associated with allergies. If you have mast cell degranulation, that's generally what you're accidentally causing to cause histone release if you have like really bad allergies. But they stain basically as opposed to acidically. So we call them basophils because they stain basically. And it's just really interesting. It was not entirely understood exactly why or how they actually do stuff, though I encountered a professor who was an expert on basophils. He's one of the few, and he was telling me that basophils are basically responsible for a really early parasitic response. But just like all these other granulocytes, so neutrophils and eosinophils, their job is just to degranulate and to do stuff. And so they do that in response to different things. And so what we're going to start to see is that there's not just one type of immune cell. There are multiple immune cells, and they have different jobs because there's lots of different things that humans have to deal with. Now, a really interesting cell type are natural killer cells. So natural killer cells do a different job. And so natural killer cells, what they do is they roam around your blood, and they look at every single cell that they encounter. So they're walking around, they don't walk, they float around, and then they encounter a cell, and they say, hey, cell, do you have MHC class 1? Which we haven't talked about MHC yet. This is the only context today that I'm going to use MHC class 1 in. But basically, it's just this protein that cells put on their surface. And you're supposed to have MHC class 1. Every cell in your body, with the exception of red blood cells, should have MHC class 1. And so we want to make sure that there aren't any sneaky spies around. And so a natural killer cell will come along and say, hey, where's your MHC class 1? And the MHC class 1 is presented, and it says, okay, that's cool. You can keep on doing your thing. But if the cell is a cancer cell, or if the cell has a viral infection, then oftentimes it will not have MHC class 1. So you can imagine a cancer cell is just growing like crazy. It's out of control. It's just not doing things right, and it forgets to put MHC class 1 on its surface. Well, a natural killer cell would say, hey, you don't have MHC class 1 on here. So look right here. So here's our cancer cell, and it doesn't have MHC class 1. And so the natural killer cell says, hey, where's your MHC class 1? And the cell says, I don't know, I don't have it. And so it is destroyed, or it's lysed. And it undergoes a process called apoptosis. That's just controlled cell death as opposed to uncontrolled cell death. Because uncontrolled cell death, like if you just, make a cell explode, like you cut it or something, that can cause inflammation. But apoptosis in general doesn't cause inflammation, because we know from just a minute ago that inflammation can be really, really powerful, and it can be really, really dangerous too. And so that's the job of natural killer cells, is to look for cells that don't have this special marker called MHC class 1. We're going to talk in a little bit about MHC class 2, which actually has a different job, but they have similar structures, so they have similar names. The last big cell type that we're going to talk about in the innate immune system are macrophages. And I'm also lumping dendritic cells into this group. So dendritic cells are very similar to macrophages. They have basically the same kind of job. Yes, they have different functions if you're an immunologist, but for this class, it doesn't really matter. Basically, a macrophage's job is macro means big, and phage means to eat. And so they will undergo phago or phagocytosis, and they just eat stuff. So they eat pathogens, they eat dead cells, they eat just basically anything that's not a living cell or that looks like a foreign cell. So they'll eat foreign things, they'll eat friendly things, it doesn't matter. Their job is to clean things up. And so they have tons of different functions. You have different types of macrophages in every one of your tissues. Those are called resident macrophages. I mean, we're talking in the liver, in the brain, in the intestines, basically everywhere has its own specific type of macrophage. And their job is to engulf these bad guys. Okay, so we've got in the green would be like the bad guy, and it's engulfing it. And then it breaks it down, and it does what we call antigen presentation. And so antigen presentation is when a macrophage, so here we've got a bacteria, it's going to eat a bad guy, and it's going to chop it up. Okay, so it does that by using lysosomes and proteasomes inside of its vesicles. And so it smashes that in there in this big old vesicle, and it chops it up, and it takes those pieces and it puts it onto something called MHC class 2. Okay, so I just talked about MHC class 1, right? Well, MHC class 2 is only for these special cells. So it's only for macrophages, dendritic cells. Technically, B cells have them, but they use them for a different job. And so what happens is the macrophage gobbles up the bad guy, chops it up, takes those pieces and places it into the cleft or this little grabby part of MHC class 2, and then it's going to head to the nearest lymph node. Now, lymph nodes, I'm sure you've heard of before. We're going to talk about lymph nodes, but first we need to talk about antigens. So an antigen is a molecule, usually a protein, that can trigger an immune response. And so in this context here, we've got our bacteria, which bacteria are really, really big compared to, like an antibody per se, which we're going to talk about the structure of antibodies later. And so part of the bacteria are these big chunks of protein, and those proteins themselves can be really large. And so what we do is we have antibodies grab onto just one little portion. We call that an epitope. And I know there's a lot of terms being tossed around here, but basically an antigen is just like a big old protein, and we want to use our antibodies, which are like our little weapons, and we use those to stick onto very specific pieces, like one little piece of that antigen, one little piece of that protein. And where that grabs onto, we call that an epitope. And so, like I said, a macrophage or dendritic cell, they're going to gobble up a bad guy, chop it up, put it on MHC class 2, and then they head to the nearest lymph node. So let's talk about lymph nodes. Lymph nodes are really, really interesting structures. And so the way I like to explain lymph nodes is that the lymph node is where the party happens, okay? So antigen-presenting cells like macrophages or dendritic cells, they're going around, they're looking up all these bad guys, they're eating bad guys, they're taking their proteins, they're chopping them up, and they're putting them on MHC class 2. And they are really excited. They said, oh my goodness, I just found a new bad guy. I need to go to the nearest lymph node because I know that's where T cells and B cells like to hang out, and that's where I like to chill with them and talk to them and explain this new thing that I just found. And so with a lymph node, that's where the macrophage goes in it. So it heads in there. The macrophages can either go through the lymph, so the lymphatic system, which basically runs alongside the arteries and veins of your body, or it could also traffic through the normal blood system. But basically, you just need your immune cells to end up inside the lymph node. And so what happens there is the key to the adaptive immune system. And so we come back to this chart here. So we have our adaptive immune system on the far right here. And the adaptive immune system, that's our T cells and our B cells. So those are cells that you've probably heard of before. And we're going to spend some time talking about them because they are the key to why you don't get sick from the exact same pathogen ever again. Now, before we get into their specific functions, we need to talk about where they come from. And so, like I said before, hematopoiesis, or the development of white blood cells, occurs in the bone marrow. And so T cells and B cells are part of the white blood cells, and so they also develop in the bone marrow. Now, the B cells are going to hang out there, but the T cells are really special. They get to traffic to a special organ called the thymus. And so I have it up here. We've got our little thymus here. I wanted to show you where exactly it is. So here is your heart right here. There's your lungs. There's your liver down there. And we've got our thymus. It's this funny looking organ that sort of sits inside the fat, or on top of the fat glob, on top of the pericardial sac. And so you can see here's our thymus. And so that's where T cells go. And so if you have your T cells and B cells, basically what we need to do is they're so powerful. They're amazingly powerful that we need to make sure that we get rid of any of them that can attack any of your antigens. And so something interesting to understand is that everything on the planet, in the universe, and inside your body can be a potential antigen, right? We're made of proteins, just like bacteria also have proteins, or viruses, or whatever. And so anything could potentially be an antigen. And so the way that your body works, the way that your immune system works, is that we have B cells and T cells that can attack and destroy any potential antigen ever. So we make every possible B cell and every possible T cell that can attack any possible antigen in the known universe. But that can include your self-antigens too. If you don't get rid of the T cells and B cells that attack those self-antigens, you're going to develop what you've probably heard of before is an autoimmune disease. So if you have a B cell that wants to make antibodies and it's attacking yourself, it's attacking a protein inside of your body, now you have antibodies inside your body that instead of attacking like a virus or a bacteria, they're going to attack one of your proteins and destroy it. So that's how you get autoimmune diseases like multiple sclerosis, where you're having antibodies that are grabbing onto part of your neurons and destroying those cells, and now you're having poor signal conduction, and eventually you die. And so what I wanted to do was talk about that training process. How do we actually get rid of these T cells and B cells that are what we call self-reactive, or the kinds of T's and B cells that would attack your antigens instead of a foreign antigen? And of course, I'm kind of glossing over a lot of stuff. Immunology is a very dense topic, and it requires you understanding like a lot of random things all at the same time, and so it gets a little complicated. I understand that. But we need to talk about that training just a little bit. So we say, all right, hey T cell, or hey B cell. So I'm just going to say T cells, but B cells do basically the same thing. So we say, hey T cell, does your special receptor that you have, called a TCR, or a T cell receptor, does your special receptor, can it recognize MHC? So we've talked about MHC before, right? That's on these antigen presenting cells like your macrophages. It says, hey, well, MHC class 2 or class 1, but we're just going to talk about MHC class 2 today. So MHC class 2, it takes that foreign antigen, right? So this peptide, this protein, and it puts it on its surface. Can you recognize that? And we ask it that, and it says, yeah, I can. And you say, okay, that's good. We want you to be able to work. But then we do a double check, and we say, wait a minute. Does that special receptor that you have that can grab onto this antigen with this MHC class 2 here, it's presenting that antigen, do you recognize self-antigen, i.e. proteins from like our bodies? And if the T cell says yes, we kill it, because we don't want it to escape into what we call the periphery, or just float around your blood, because that could accidentally cause an autoimmune disease, and we don't want that, right? And so that's the process of positive and negative selection. That's called central tolerance. If you take an immunology course, either with me or someone else, we end up talking a lot more about this. But basically, you just want to make sure, do your T cells and B cells work right, and are they going to attack you? And if the answer is, do they work right? Yes, we want to keep those ones. But do they attack you? That's a bad thing, because we don't want them attacking you. And so on this chart here, it's obviously complicated. We're not going to go into what DN, that's double negative or double positive, like all this stuff. We don't really care. We just know that our T cells and our B cells, we get rid of the ones that attack you, but we keep the ones that work pretty well to attack foreign antigens. So now we're at the point where I need you to pay close attention, because we're about to get to the main crux of how the adaptive immune system works. And there's a lot going on this chart, but I promise I'm going to take you through it. So when we have a foreign antigen that's encountered, so that could be a bacteria, a virus, a parasite, something like that, your antigen presenting cells, so that's like your macrophages, your dendritic cells, they're going to find this bad guy, they're going to gobble it up, and they're going to chop it up, and they're going to present it on MHC class 2. And so that's what we're seeing here on the left here. Okay, so it's gobbling it up, it's going to present it on MHC class 2, and it heads to the nearest lymph node. Now, at the same time, a B cell, so those are cells that are going to secrete antibodies. Okay, and we're going to talk a little bit about antibodies in a second, but B cells, they also do kind of a similar process. And so they use their antibodies on their surface. We call them B cell receptors, but they're just antibodies. And so they've got them on their surface, and those antibodies, if they recognize this foreign guy, so this bacteria right here, if they recognize that, they will gobble it up and chop it up and present it on MHC class 2. Now that's the exact same thing. So MHC class 2 over here, MHC class 2 over here. Yes, those are the same things, but their job is a little bit different. We're going to get to that in a second. But if that happens, so your B cell has gobbled up the bad guy, taken it up, presents it on MHC class 2, it also goes to the nearest lymph node. Now, T helper cells, and we're going to kind of skip over T killer cells today, but T helper cells, those are just kind of wandering around the entire body, and they like to hang out in the lymph node too. So if you remember, lymph nodes are where the party happens. And so the lymph node is where all three of these cell types are going to come together. We're going to have our macrophages come together. We're going to have our T helper cells come together, and we're going to have our B cells come together. Now, we haven't really talked about T helper cells yet. So why are they called T helper cells? Well, that's a great question. And so what the T helper cell is going to do is it's going to act as a check and balance to the amazing power that B cells have. And so what I want you to do is look here. So we've got our macrophage here. In this case, it's a dendritic cell, but it doesn't really matter. So we've got our dendritic cell here, and it's got an MHC class 2. So you can see it in blue here, and it's presenting an antigen. So in this case, it's a little green antigen on here. It's showing that to our T helper cell. So our T helper cell, there's millions and millions of them, but only one, I'm exaggerating just a slight bit, but only one of them actually escaped the thymus. Remember, we got rid of all those self-reactive ones, the ones that can attack you. So this one has a special T cell receptor right here that only recognizes this specific antigen. And so if that happens, if this macrophage shows this specific antigen on MHC class 2 to this T helper cell, which has this very specific T cell receptor, then magic happens. This T helper cell becomes activated. And so what it's going to do is it's kind of like it got super excited and it says, oh my goodness, I just talked to a macrophage and it just told me about the coolest antigen I have ever seen before, and I need to go tell a B cell. And so the T helper cell, then it's in the lymph node, right? And so it starts looking around and says, hey, we're having a big party here, right? There's macrophages here, there's other T helper cells, there's B cells. Hey, I'm going to go check with each of these B cells and see if they also have this exact same MHC class 2 with this exact same antigen. And so that's what we're showing here. So we've got this T helper cell, he's going along, and I just kind of changed what it looks like, but there's this little T cell receptor, same thing. So it just says, hey, I'm activated. Do you have an MHC class 2 with that exact same antigen in it? And if the B cell encountered that same thing, so they're gobbling up the same bad guy. So that B cell came into the lymph node and says, hey, I also have that exact same antigen on my MHC class 2. Hey, that's pretty cool. And you're an activated T helper cell? Oh, snap. That's when the magic happens. And so the B cell was waiting. It was primed. It was ready to go. B cells make antibodies, but it won't do it until it gets some help. And that's what T helper cells do. So T helper cells have to get activated by macrophages, and then the B cell has to encounter that activated T helper cell. This is so important because if a B cell could just randomly get turned on and start making antibodies, well, what if it accidentally started attacking, yourself, and then you have an autoimmune disease? That's really, really bad. The adaptive immune system is so powerful. It's very, very energetically expensive as well. And so just think about, like, if you've ever had a cold, you're really tired, right? Well, that's sort of a symptom of your immune system working hardcore overdrive to make tons of B cells and tons of T cells and tons of neutrophils and tons of macrophages. That costs a lot energetically. And so we don't want to just randomly turn on the adaptive immune system because it's so powerful. And so what we want to do is have a nice check and balance. And so you can't get a B cell response without a T helper cell telling it to do that. And you can't get a T helper cell activated without a macrophage telling it to do that. And so we have this nice check and balance here. Now, I am ignoring T killer cells. That's something that we are not planning to talk about in this class. Basically, what they do is go through a similar process that the B cells go through, except they go around and look for virally infected cells. And so we're not going to focus on that today because it's more important that you just understand the general idea of how we're generally going to attack like a bacteria. And so I have another diagram here. It's a little more simplified. And so here's our macrophage, just to kind of go over it again. There's MHC class 2 with this antigen. Okay, so from like a bacteria or virus or whatever, and we tell our T helper cell, hey, do you recognize this? And maybe 1 out of a million actually do recognize it, and that becomes activated. And then that T helper cell is going to go look for a B cell. So this is that same T helper cell that got activated, and it also has its same T cell receptor, and it says, hey, B cell, do you have this exact same MHC class 2 with this exact same peptide? And if it's true, if it does, then that B cell becomes activated and begins secreting antibodies. So let's talk about antibodies. So antibodies have 2 chains. So they have quaternary protein structure. What does that mean? Well, that just means they're multiple polypeptide chains that are stuck together. And so we've got some disulfide bonds here that are holding them together. And we've got a heavy chain. Okay, it's really long. And there's 2 of those. So we have 2 heavy chains. And we have 2 light chains, heavy being, they're just literally heavier. They're a lot bigger, so they weigh a lot more. And then we've got our light chain, and it's lighter. So it just doesn't weigh as much. And so there are 2 parts to each of those. So we have our variable region, and this is the business end of the antibody. So this end is the part of the antibody that will grab onto a bad guy, or just basically any kind of epitope on an antigen. There's also the constant region. So you can see it's listed in C here. That's what these C's stand for. And the constant region is the region that stays constant. And so what does that really mean? Well, this region is where a macrophage or a dendritic cell or a neutrophil might grab onto an antibody that's bound to a bad guy. So let's say that an antibody has grabbed onto a virus, like coronavirus or something, and it's grabbed onto it, so we know it's very specific. And then macrophages and neutrophils and stuff will have receptors that grab onto this end. And so that can really help the macrophage to pull in any viruses that get attached to these antibodies. Now, there are different kinds of antibodies. There are 5 specific classes of antibodies. There are subclasses of these, but these are the 5 big classes. And so we start out with IgD, which isn't super common. It's really early on. And then you have IgM, which is the first one that we generally make when we have a bacterial infection or a viral infection or whatever. It's the first one you make. And you can see it has pentameric structure. There are 5 antibodies stuck together, which is really cool. And then you switch over to IgG. And IgG is generally the main type of antibody that you're going to encounter. This is really, really special. It can cross the placental barrier. It can cross in breast milk. And so IgG is probably the main antibody that you're ever going to encounter. Though there are some specific subtypes. So if you're fighting a parasite, like a helminth or a worm or something, then you would switch to IgE. Oh, and if you are trying to protect your mucosal surfaces, then you might have some IgG floating around, but you can also make IgA, which has a dimeric structure. There's 2 of these, and so there's 4 binding sites. And IgA is what you actually secrete onto mucosal surfaces. And so we've talked about mucosa earlier. And so like in the vaginal compartment, in the intestinal compartment, in your throat, in your lungs, you secrete IgA, and it's at that first line of defense. So inside the mucus, which is really sticky, you can also have IgA, which can bind onto bad guys. Now I have a chart here which talks about a little bit more about their specific structures, their specific names, so mu, gamma, alpha, epsilon, and delta, and just how big they are and how much of it is normally just this percentage. So like mostly it's IgG, but you got some IgM in there. Sometimes you have IgA, at least on those surfaces, and they have different jobs. But basically, you want to concentrate on IgM and IgG. Those are the 2 biggies. IgM is the first one that we generally make, and then we switch over to IgG because it's much more specific. The binding regions for IgG, the variable regions, are more specific to a specific antibody or to a specific pathogen than those are for IgM or IgD. Now we're going to lean heavy into IgG. That's the main type of antibody that we want to make. And generally, IgG has a variety of functions. So I have them right here on this chart. And so generally, they want to neutralize bacteria or viruses. And that just means they just stick to stuff. And if you have a bunch of antibodies and you're a bacteria that they're stuck to your surface, then it's a pain in the butt to try and get around. And so it's neutralized. And then what that can also do is it can opsonize that bacteria. And opsonization is what I mentioned about earlier, where macrophages or neutrophils will have special receptors that can sense antibodies, and it helps them grab them and pull them in. And so macrophages like to phagocytose, right? And so if a macrophage senses an antibody that's bound to a pathogen, it can use that through opsonization to help phagocytose that bad guy and destroy it. There are a couple other ways that these work. So antibodies can help trigger natural killer cells to blow up cells. They can help trigger T killer cells to help blow up cells. They can also influence complement. So complement is something that we're not going to talk about today, but basically it's part of the innate immune system that can cause holes to occur inside of pathogens or any kind of cell type, or it can also sort of coat the bad guys in proteins that cells recognize as something that should be destroyed. And so that can also trigger phagocytosis, which antibodies can also influence that. So not only can they opsonize bad guys themselves, but they can cause complement to occur and then opsonize. But for the most part, neutralization and opsonization are the 2 big ways that antibodies actually damage pathogens. The very last thing I want to do today is bring us back to the original idea that the body can remember the pathogens that it's encountered. So immune memory is so important. And the idea of immune memory is actually pretty straightforward. So every time you get exposed to some kind of antigen, and so that could be some kind of infection, a bacterial infection, a viral infection, coronavirus, whatever it is. It could be a vaccine. So when you get that first exposure, it takes about 2 weeks to actually encounter the antigen, have a macrophage gobble it up, present that to T helper cells, which then can trigger B cells to actually make antibodies, and that takes about 2 weeks. You start making IgM first, and you end out with a little IgG, and hopefully you get rid of the virus. Now, when you get that second exposure, or a tertiary exposure, or just any kind of subsequent exposure, what happens is you can skip that whole step of having to go from a macrophage to a T helper cell to a B cell. You can skip that step. And so that B cell can just be triggered immediately. So we have memory B cells and memory T helper cells that can then just immediately be triggered. And so within a day or two, they can ramp up their production hardcore. So B cells start making so much IgG, and they make it so much faster. And so they make way more IgG, so the response is bigger, and they make it much, much more quickly. And so that's what I'm trying to imply here with this chart, where that primary immune response, or that first immune response, yes, you get rid of the infection, but any subsequent infection is much quicker and much more powerful. And this is why vaccines work, because vaccines artificially give you your primary immune response. So the next time you actually encounter a pathogen, it's not this response, it's this massive response that can hopefully keep you from getting very sick, or at least not having very many symptoms. And that, my friends, is the immune system. Now, next week, we're going to be talking a little bit about viruses, which are going to trigger lots of these things. And we're going to be talking about vaccines a little bit. And vaccines are super, super cool.